Hello, everyone. I'm Jessica Carmen. I'm the Director of Business Development for MaxCyte's cell therapy programs. I'm delighted to talk to you today about our enabling technology and our proprietary therapeutic products. So first, disclaimer. So as I mentioned, I'm going to talk about our enabling technology, which is accelerating the development of our partners' ex vivo gene therapy programs through their clinical developments, product and clinical developments, as well as our own proprietary therapeutics, which I'm excited to talk to you about today, where we're driving innovative cell-based medicines into the clinic ourselves. First, about our technology. Our technology is our patented flow electroporation. Our entire technology was designed with cell therapy in mind. It was always the intention to use our technology to deliver molecules into cells for the purpose of delivering those cells into patients. To that end, our entire technology is GMP compliant and suitable for use for patients. It is a non-viral technology, so it's a way to introduce genes, molecules into cells without the use of a virus. And we have collaborators with academic and commercial pioneers around the world, and we have a broad portfolio of IP on our technology. Our technology can address multiple different types of products. Across the top, the engineered cells or the ex vivo gene therapy, those are the cells we're primarily talking about today. This is the types of products in which genes are being delivered via cells. We talked about this in great detail in the workshop this morning, if anyone attended the gene therapy workshop. Additionally, our technology can also address in vivo gene therapies in the sense that our technology actually can be used to engineer viral vectors. Our technology also can be used for therapeutic proteins such as monoclonal antibodies. And also our technology can be used to engineer cells for screening of small molecules. For most of the cell types that people are interested in working with in this space, we can see greater than 90% efficiency, as well as greater than 90% viability for cells that are processed with our technology. The most common types of molecules that people are loading into cells are mRNA, but also DNA plasmids, siRNAs, proteins, or any combination thereof. Any type of cell that you want to work with, we can address with our technology. So hard to transfect cells, primary cells, cell lines, all of it. And the range of cell numbers, it would be from 500,000 cells all the way up to 200 billion cells. This is the largest scale that can be addressed in this space. And everything, as I mentioned, is clinical grade and suitable for use for treating patients. So everything is sterile, single-use, disposable. And also everything under quality systems is GMP compliant. This is the family of our instruments. On the bottom, the red instrument is our GT, our gene therapy instrument. The range for the GT goes from 500,000 cells all the way up to 20 billion. And if you need 200 billion, then that can be addressed with our larger instrument in the middle there, the VLX. And then the instrument that's shown at the top, the STX, that is the instrument that can be used for viral vectors, therapeutic proteins, and small molecule drug discovery applications. Our buffer and our consumables are all medical grade. The buffer is a chemically defined GMP compliant buffer with no human or animal origin components. The buffer is made in a GMP facility suitable for use for humans. Also, our medical grade disposables have sterile weldable tubing. And we have a device master file with the US FDA that is cross-referenced by our partners that have products in clinic for which our technology is supporting. And additionally, we have one product that is on the market in Japan. So here's a list of some common applications for which our technology is being used. As I mentioned, viral vector production, CAR T-cell engineering, dendritic cell immunotherapy, such as the Japanese product that's on the market, engineered stem cell therapies, directed stem cell differentiations, as well as gene editing applications. At MaxCyte, we believe that the truth is in the science, and so we are proud to demonstrate the publication record of our scientists and our collaborators. So an example I'm showing here is our large-scale lentivirus bioproduction using our technology. This was a collaboration back in 2012 demonstrating the use of our technology to generate lentivirus from suspension cells. Also, one of the first publications in the field of CAR, actually, back in 2011. This is the demonstration of our technology for delivering mRNA CAR into T cells, showing that the mRNA CAR can be shown through at least 6 days post-administration, as well as also effective up to 6 days with the effector target killing. And most recently, this is a paper that was published earlier this year with our collaborators at the NIH looking at a monogenic disease called X-linked CGD. In these patients, they have a mutation in the NADPH enzyme, which renders them unable to sufficiently fight off bacterial pathogens. And so with our collaborators, we introduced CRISPR-Cas9 as mRNA into patient cells to correct the mutation, and then we administered those cells to skid mice and find that the cells were able to engraft and survive for as much as 5 months after engraftment. And the scientists at the NIH were excited to see that the level of engraftment and persistent was clinically meaningful, meaning that the amount of cells that engrafted and survived would be sufficient to actually have a clinically meaningful result in patients. So these scientists are moving forward with this study, and we're really excited to see what they do next. So the benefits of our delivery platform: it is universal, non-viral, it is very robust, consistent results every time. You can work with your appropriate cell type to maximize your results. High efficiency and viability with just about every cell type and molecule we've ever tried. And the scalability is fantastic. So everyone can work out the conditions in small scale and then easily translate to large scale without having to reinvent the wheel as they scale with the technology. And a clear regulatory pathway with associated support. So now I'm excited to talk about our proprietary cell therapy, our KARMA cell therapy. This is chimeric antigen receptor that is delivered as mRNA. This is an autologous cell therapy in which fresh PBMCs, this is unexpanded PBMCs, are administered the mRNA using our enabling technology. And our platform right now, our MCY M11, is our lead candidate. And this is addressed at mesothelin-expressing solid tumors. And we also have MCY C55, which is CD23, targeting hematological malignancies. And we have other products in the pipeline as well, which we're not prepared to talk about at this time. All of these products are proprietary to MaxCyte. Some key attributes of KARMA: it does not require the use of a viral vector. It is non-viral. It uses our enabling technology. It permits the prospective management of on-target off-tumor due to the transient expression of the CAR, and it enables the development of CAR therapies for solid tumors, and it has a much reduced manufacturing complexity compared to CAR-T. So here's a glimpse of the pipeline. The MCY M11, as I mentioned, is our lead candidate. It's going to be addressing peritoneal cancers, and it's marching toward the clinic. Right behind it, our MCY C55 for AML is on the heels. And then we have a few others that we're working on that we're not disclosing details about at this time. And we get asked a lot, what is the difference between KARMA and CAR-T? So here, just sort of a little side-by-side so you can see what some of the differences are between our KARMA and what everyone is familiar with as CAR-T. So we feel that we have lower off-target toxicity due to the shortened persistence as compared to the CAR-T. Also, much more rapid turnaround to the patient due to that simplified manufacturing process. It's virus-free, and therefore we believe that is a lower toxicity for the patients. Also, we've noted that for a lot of people in the space that are working with virus, that there are delays in their entire clinical timeline due to waiting for virus to be manufactured at CMOs for GMP virus. That delay is not present in the product development timeline for our products. So we think we'll be able to get through our clinical development a little more quickly because of that. Also, the multi-dose administration of the product will allow for more control of efficacy and safety. And then finally, we believe that preconditioning will not be required for solid or hematological malignancies. That's MaxCyte. So thank you.